We do not know the ancient phenotype of Rubisco, phenotype the behavior. In fact, we do not know anything about any behavior of anything ancient, if you think about it. Right? And that's what I think makes this kind of study terrifying and exciting at the same time, that we are dealing with a lot of unknowns. So what we do is we take what we see today and try to map it onto the past. We use today as almost a key into the past states. Welcome to tonight's talk. I will be talking about the research that interests me the most, understanding ancient life. So I'm very interested in understanding what happened in the past. How did ancient cells look like? How did the first cell look like? Or you can even get close and have an idea of how these cells behaved or how the molecules that compose these cells look like. I am an astrobiologist. Has any of you ever heard of astrobiology? Astrobiology is a relatively new field interested in understanding past, present, and future of life on Earth and elsewhere in the universe. And here are some questions that are interesting for astrobiology as an aspect. Interesting aspects for astrobiology, but I should stop this down. All right. So Mars Curiosity rover practically is the first astrobiologist that got to perform experiments on Mars. If we were to send a geologist on Mars, he would probably do, or she would probably do what Curiosity does, not including the laser coming out of his eyes. But I would say that Mars project is one of the first projects that included astrobiology as one of the mission objectives. So that's exciting. Next to that, we have the recent Matt Damon, who is the first fictional astrobiologist who got to perform experiments on Mars. So here you can see that as a botanist and as a geologist, already you can see the depth and interdisciplinarity that the field covers. So we have a lot of different, we have chemists, we have biochemists, we have astronomers. It's a very interdisciplinary field, and that makes it very exciting. So if you don't have the chance to go to Mars or this week, what can we do as astrobiologists here on Earth? So I'm interested in understanding the origins of life, which is a very big component within the field. So what we have is life we see today, right? So as far as we know, it occurred once, at least once, here on Earth, and we have the biology that we see now. And in the field, there are two ways we can explore how life occurred and happened to be the way it is. We summarize these as bottom-up and top-down approaches. The bottom-up approach is about accessing the non-life components. For example, nucleic acids, DNA, amino acids, or maybe cells that we can even produce from that encapsulates these non-life components. And then from there, can we create life, starting from non-life? The other component is life, right? So we can take what we have right now, the cells or ecosystems, or even components that form cells, like such as here shown ribosome, and then one by one, try to understand how can we track our way back into the past. So it's a really backwards from today kind of approach. And complementing this, we have geology, right? We got the rocks. Rocks tell us a story about the past and the story of the Earth. And we got other more exciting, perhaps, components. For example, marine ecology. If you're not satisfied with what you see on surface, we can go subcellular or subsurface levels. We can go underneath the oceans, or even Antarctica, and here shown a pH low lake in Spain. So there are different environments that we can explore elsewhere on different places than the lab, I would say. So in this talk, I'm going to focus on the bottom-up approach biology, what we have right now, the cellular biology, and combine it, a new approach that will combine it with geology. All right? So our goal is to reconstruct the past, and we ask, can we reconstruct the past? And overlap geology and biology in a novel way. So what does geology do? Geologists use fossils. Fossils are the preserved remains of once living organisms. And the fossilization, it's a rare event, right? So there are problems or maybe shortcomings of geology. We need fossils so that we can analyze the past. However, as I said, fossilization is a rare event. And the rocks that we have right now are not necessarily naturally selected, right? Earth is also moving all the time. It's tectonically active. So we have to find these rocks, and they are not necessarily here because for a reason, unlike biology, which I'll come to in a minute. And small organisms may not be preserved well. And the same thing applies to older fossils. The older the fossil is, the chances are it's going to be degraded by a geological activity. So we are limited in the sense of our way back into the past. And then we have biology, right? We got the genome, we got the DNA. If you think about it, it is a fossil on its own. It does contain information about the past. It's been evolving. Evolution has been there. It's an ongoing process. It's happening right now. And the information that is conserved on the genome is majorly naturally selected in general. However, the DNA is overwritten. Past happened once, and we cannot repeat it again. So what we have is what we see right now, and we got to do something with it. Here is an example of how we can use DNA to understand relationships between currently existing biological organisms. And I'm showing an example for dogs here. So we got different breeds. And by creating these trees called phylogenetic trees, we can understand the relationship between different biological taxa. Right? We can look at their ancestry, when the splitting occurred between breeds. So let's zoom in a little deeper. The question that we can answer, the questions that we can seek using these trees, is that which extinct species evolved from the same ancestor? And how were ancestral traits modified, right? What are their roots? Where do they go? When did they split? Are they related? Are they closely related or not? Looking closer to trees, getting closer, here's an example, right, to explain you. Yeah. So I am the immigrant he's referring to, by the way. So if he's selected, I'm not coming back to the US. I mean, I can't because, anyway. So yeah, it's... So here are two very sad organisms. And they share a common ancestry, all right? So this is what we call as nodes when we read the tree. And then we have another common ancestor that is shared with this overall group. Here's another node. The branches here represent the time, right? So the longer the branch, the longer that the history is. And the nodes tell us something about the ancestral state. So we can trace back the nodes, and we can say something about the state of the ancestor. And this happened, the split between the cats and human happened 80 million years ago. Again, human is questionable here. So we can then ask the question, can we use these phylogeny trees as a way to travel back in time and perhaps even infer what was going on in the node with that time point, right? Not just get a snapshot of the time and when the split occurred or get an estimate of when that happened, but get information about the time period and also the character state within that time period. This was an idea that was proposed by a chemist and a physicist. This is how science is beautiful. The interdisciplinarity can help you. A chemist and a physicist is getting together trying to help biologists, and it works most of the time. So they have this paper. They're published in 1964, one of my favorite papers, Molecules as Documents of Evolutionary History. So here they proposed a thought experiment. And they said, well, if we take currently existing sequences, the DNA sequences, can we treat them just like, instead of DNA sequence or amino acid sequence, can we just treat them as life's language, words, and then trace back the ancient word states? So about 30 years later, a chemist realized this paper and then implemented this idea, and for the first time used phylogeny trees to reconstruct and resurrect an ancient protein. So now I'm going to walk you through what's going on. So this is how Hollywood travels back in time. As scientists, things are a bit more fun. This is our version. We don't need any flux capacitor. If you were to understand an ancient state of a language, you would take your favorite language and then study the cultures that use that word or the language, say the words, sentences, and then try to understand something about the past state of the cultures that use that language. This is the Turkish alphabet, and this is a biologist's alphabet. Instead of letters, we have amino acids, we have nucleic acids. So we can treat the words, or the letters that compose the words, the same way. Here is the word snow, very relevant for New England. I am learning. So these are the different cultures that use this word, all right? So we got snow here in English, we got nieve here in Spanish. All these different cultures have this word. So what does this tell us at first? That these cultures lived in a place where it snowed. They needed this word. So their environment can be inferred from the words that these cultures use. And then if you trace back this word, we're going back to 3000 BC, we get this word as the ancestor of the extant word snow. So we can then apply this to biology, right? Instead of a Germanic or Romance culture, we have bacterial cultures. We got proteobacteria, cyanobacteria, and instead of the word snow, these are the words that are composed of amino acids. And each of these words share a common ancestor, right? So let's start from here. We got L, H, V, P, K. These are all the letters that represent amino acids. And we have the descendants from that ancestor. And we can trace back all the way to the ancestor, the common ancestry. That's exactly what we do when we use phylogenetics for our studies. We take currently existing DNA or protein amino acids, and for our favorite protein, we get sequences from a human, a crocodile, or a plant, or if we want to keep it more specific, we take it from different species of within bacterial species, different cultures, different organisms. And then we align these sequences, and then we ask ourselves if say in this position 1, if all of them had a letter C, the ancestor also most likely to have a C in that position. So one by one, we apply this maximum likelihood technique and then reconstruct the ancestor. Nowadays, we send this to companies who do this synthesis step for us. It is $1 per amino acid. And then we take the construct that they ship back to us and then resurrect this protein inside the lab by following very traditional molecular biology cloning techniques. And at the end, we understand its properties by doing basic biochemistry or whatever the assay that we have to do in order to understand that protein. This is how it looks like. I'm showing a very simplified version. Usually we use 20 to 30 different sequences. It can be more depending on the protein that you would like to use. And we perform alignments, lots of alignments. And nowadays, this is actually going to be very soon publicly available. If any of you would like to resurrect a protein, you can use this system, this online program called Phylobot, a friendly robot that automates your phylogenetics, and I am not endorsed for this. It's by my collaborator, Victor Hansen-Smith. It's phylobot.com, and if you would like to give it a try even nowadays, you can. So, I would like to use this ancient gene resurrection system to understand geological past. This is me as a TARDIS, and this is Victor and Zach. Zach is here. We have a geologist and bioinformatician that I'm collaborating with for this project. So, let's take a look at the history of biological and geological life on Earth. We have now, we are here, the formation of Earth. So you're looking at a 4.5 billion year timeline. I'm going to highlight several important time periods. One is the Cambrian explosion. It is not a real explosion. Scientists like to use these dramatic words. It's just an increase in animal fossil, amount of animal fossil within that time, about 540 million years ago. We got the rise in atmospheric oxygen about 2.2 billion years ago. We've got our first evidence as a fossil that life existed, like that shows that life existed around 3.5 billion years ago. And an isotopic evidence for life, around 3.8 billion years ago. suggesting that life is as old as 3.8 billion years. So what does isotopic evidence for life mean? So we have the living organisms that are selective in the carbon isotopes that they use. We got two different carbon isotopes, carbon-12 and carbon-13. What we see is that the living organisms have a higher carbon ratio, isotope ratio, compared to non-living states. That's what it is all about. Here is the example, the fossil that I was talking about. That's about 3.5 billion years old, found in Western Australia, the oldest fossil. So you're looking at your great-great-great-great-great-great-grandfather or mother. And isotopic analysis, this time from a fossil, that suggested that carbon fixation was as active as as old as 3.8. So we have two evidences for carbon isotope effects that suggested that life was active back then. Going back to the timeline, however, I would like to point out that there is a big debate in the field discussing did life actually start around this time, right? So we got the late bombardment, the heavy bombardment, the bombardment of Earth and Moon with meteorites and asteroids, right? So what did that do is that it really destroyed the surface of the Earth. So some scientists think that life existed before the late bombardment. However, because of this heavy shower, life vanished. And what we see could be a second life that started on Earth again. Some believe that life didn't vanish because of this late bombardment. And the fossils we see simply they represent a generation that is older than them. And some think that, no, these are the first evidence for life, and life didn't start until the meteorite shower. A recent paper, this paper came out about 2 months ago, suggested that life is in fact older than what we think it is, how old we think it is, as old as 4.1 billion years, right? So this paper studied a zircon, and they found these dark spots on this zircon mineral, and they measured carbon activity in these dark spots. And they inferred this information as life being present as far as the carbon isotope activity goes, as old as 4.1 billion years ago. This is a very recent study, and what I see in geological conversations is that it's not accepted. It's far from it. It created a big debate. But I think if you're interested in these things, this is an argument to keep an eye on. We'll see what will follow these studies. But let's focus on the isotope records. Here, I'm basically trying to show you that isotope fractionation values are very important, that we directly infer this information for the evidence of life. So here we're looking at a different timeline. We're time 0 today, all the way to the past, and different records that shows different evidences for life's presence. We're focusing on the isotope record again. And in general, you see, as we go back in time, we see more spotted information, which is also I was talking about in the beginning, that how it gets very difficult when you're dealing with really old time frames, right? So for isotope record, it's the same. We have big gaps. We have no idea what happened here. That's kind of cool. Yeah. But the question is, what are the biological components that we think is responsible from generating this biogenic isotope fractionation value? Why do we see this? Why do we look at this? What causes this? So this is one of the enzymes that is responsible from creating this carbon signature, and it is called rubisco. And if you cannot remember rubisco, please remember ribulase 1,5-bisphosphate carboxylase oxygenase. And remember that chemists are awesome, naming things. So rubisco does fix carbon dioxide. So it binds to carbon dioxide, and it fixes it on sugar. And it is involved in photosynthesis overall. It's a very important enzyme. This is the reaction mechanism that rubisco is involved in. Here is the stage that it plays a role in. It's essential for the fixation of atmospheric carbon dioxide. It is very slow, and it's a very confused enzyme also. It cannot differentiate carbon dioxide from oxygen, so it binds to oxygen every now and then. It is probably the most abundant protein on Earth and contributes to overall carbon balance. So it's very cool. A lot of studies try to improve its activity. Lots of directed evolution studies or synthetic biology studies that focus on just making faster Rubisco. So we don't quite know what makes it too slow, but it is what it is. So, can we reconstruct the ancestral Rubisco? You see where I'm going with this. I want to go all the way in the past and try to understand what would the ancestral Rubisco's carbon isotope fractionation value would be. And if you're looking for the answer, I'll see you in 2 years. It would be funny if I just left right now. I'm not leaving right now, but anyway, just drop the microphone. Yeah. So you're looking at I would say, a protein selfie. Okay, so this is what a protein looks like, or it looks like this. But this is what a biochemist sees when it looks like this. All right, so what can understanding an ancient protein tell us about geology? Right, so we're looking at this and we're trying to make inferences about Earth environment, about Earth. Okay, so how do we do that? These are the boring key questions, perhaps, but this is how we get the grants. All right, so the ancestral Rubiscos, we're trying to understand, as I was saying, how they would contribute to the carbon isotope fractionation value. How far can we trace these values? And if we can't, then why not? How did the ancient state of this protein even look like? And as I was mentioning, there's a rise in oxygen around 2.2 billion years ago. How did this protein behave then? If you recall, I said this protein is very confused. It doesn't differentiate between oxygen and carbon dioxide. So what was the Rubisco back then doing? And what was the most ancestral, the ancient one, the most ancient one behaving like? These are the old questions that gets you into studying ancient life, basically. If you're interested in this, then you're welcome. So the problem statement. We do not know the ancient phenotype of Rubisco. Phenotype, the behavior. In fact, we do not know anything about any behavior of anything ancient, if you think about it. Right? And that's what I think makes this kind of study terrifying and exciting at the same time. That we are dealing with a lot of unknowns. So what we do is we take what we see today and try to map it onto the past. We use today as almost a key into the past states. Not only we would like to understand this protein's behavior biochemically, but we would like to design organisms that contain this ancient gene in its genome. So not just inside a tube, but also an organism that functions, being dependent on this ancient gene. So here's how the ancient sequence phylogenetic tree, the phylogenetic tree of Rubisco looks like. We generated this using Phylobot, and this is the simplified version of the tree. We have the currently existing sequences. We obtained this from plants and bacteria. And we have every node here that represents an ancient state. Here are the current structures of existing Rubiscos. Basically, I'm showing different protein structures obtained from different organisms. They are overlapping because they look more or less the same. But importantly, Rubisco functions very differently, actually, from organism to organism. It has different kinetic values, it has different behavior, but it looks the same. I collaborated with Nick Vocalhide from Colby College for this project. And what we did is we used the current structure of Rubisco and modeled the past structure. So here is one. Here's another one. Here's another one. And as you can see, the structures don't change as much. So, like that, all right, one second. Okay. So what we do is that we try to understand not only how these ancient proteins look like, but also how they move. All right, so can you, oh yeah, it's a 5-second movie. It's so exciting to me. Could you please, yeah, just click it. Yeah, there you go. So we model this, and I can watch this whole day. It's very exciting because not only you get to get a peek of the past state of a currently existing molecule, but you also model how it may behave. And you don't ideally want it to move like an octopus. You know, you want it to be kind of put together. That's going to be very helpful for the subsequent biochemical steps. It will presumably, hopefully, will help me to understand its function so I can actually first work with it, isolate it, purify it. So I want it to actually look not too dramatically different from what I currently have, so that I can apply what I know for the modern version to the ancient one. Well, based on what we see right now, based on our sequence analysis, we think that this protein has been very fine-tuned. It didn't change as much throughout its evolutionary course, at least as far as the structure goes. But for protein, like in life, the looks aren't everything. So we need to look at the actual function, and we need to look at what it does to understand its functional evolution. But now that we reconstructed the ancient rubiscos, as I showed you, we have their models. We got a peek of how they look like. We are going to be mapping the properties with historical geological events by synthesizing these inside the lab, bringing back to life, and then understanding the biochemistry of these molecules and then mapping them with isotope fractionation values that geologists identify. So we are providing another data set by going back in time to rubiscos. We create this ancient data set for the isotope fractionation, and then we're going to map it to what geologists calculate. I got into studying ancient genes and proteins because my main interest was to understand whether evolution is a repeatable process. There's this notion in biology that we refer as the tape of life. But the idea is that everything that took place on the surface of the Earth, just imagine that it was recorded on a tape, and you don't know the tape. If you don't know the tape in this room, I will be very sad. But tapes were these things that we used anyway. So if everything is recorded on a tape, and then we were to rewind the tape, basically destroy everything on Earth, and then replay evolution again, would we even be here? Or would the life we see around us be different? If so, how? And why? So that to me is a very profound question. And even though I acknowledge that what we do is not necessarily going to take us to the absolute truth, I think it's a good attempt to understand what may have happened. So the goal is to not only to reconstruct these genes, but also give them a second chance by evolving them in the lab, and then follow their evolutionary pattern and compare them with what we currently have right now. Which brings me to, I have 2 slides on this, that how I really appreciate this effort here. I know this is not a big problem for Cambridge area, but this is the public acceptance of evolution based on a study, and this is where I'm from, and this is US. So when I came, things really improved for me. Yeah, so Greece next. But it is, I was thinking, this is very interesting because US invests so much, money and invest so much resources on understanding evolution. Yet, Turkey, we proudly do not. So we deserve our spot. But I don't know about what's going wrong here. But so what happened is this is when I understood what may be going on. So another part of my study, based on a paper I published, got picked up by a newspaper, and then it got kind of viral around the world. So this magazine put my picture, and then next to Jurassic Park. So because it's very relevant. So and they called it what could possibly go wrong. So, and they were referring to my career, I think, which terrified me. So what happened is, this went on viral, and then people started putting these comments on, and this is how I spent my lunch breaks. And then I'm getting these emails, of course, that say similar things, that smiling woman should be crying and terrified about the procession of our new ape masters. Or, I have seen this movie and didn't end well, or isn't this how AIDS was created? So if you are leaving this talk with these questions in mind, I clearly failed, and I didn't do a good job explaining my work. So please come find me, I can explain. But my students back then gave this dinosaur to me, and so instead of 1 smiling woman, now we have 4 smiling women. That's more terrifying. So with that, I would like to end. I have these three gentlemen working with me on the Rubisco project, and I have a lot of undergrads and collaborators that work with me in different aspects of this project. This work majorly was funded by NASA and now John Templeton Foundation. And if you're interested, if you've got a bachelor's degree or a master's degree or a PhD, I'm currently hiring, so I'm shamelessly putting this ad here. So give me a holler. All right, thanks.
My question is on Rubisco, the ancient version versus the newer version, or what we have right now. Does the codon usage differ that much, or is it all the same?
We codon optimize these things before we put them inside the cell.
But by themselves, the sequences
alter
a lot.
So we see a variation. The younger ones are identical up to 90-95%. And as we go older, it goes down all the way to 40%.
For Rubisco, over time, I think it gets a little bit more efficient, right? So what's in the mechanism that makes it still want to bind to oxygen and carbon dioxide? Sort of like they have the same affinity you think it would optimize by now, right?
Yeah, that's a very good question. So based on the structures and the sequence identities that we see, we don't think that the activity was that different, the activities of the ancient ones. And, but we don't really know because I haven't done the biochemistry essays yet. Does that answer your question?
Yeah.
But we do not know. Like that's a very big question for the Rubisco field, how to improve its activity. And we think that perhaps maybe going back in time, like using these ancient ones, if we see a faster working enzyme, that would be very interesting. But short answer is we do not know what contributes to its kinetic activity yet.
500 million year old gene, what did it do? Was that the oldest gene that was ever been reconstructed or isolated?
No, so that gene was functioning in the ribosome. So the protein making machinery of the cell. Basically, this protein was delivering amino acids to the ribosome. It's called elongation factor. It's actually, it's older than 500. It got as 500 million year old to the media, but it was about 700 million years old. And so it was right before the Cambrian explosion. And it was not the oldest one. It was actually the youngest one. That's why I picked it. It goes all the way to 2.8 billion, 3 billion year old. So basically, we are proposing that elongation factor, with its essential role in the cell, is as ancient as the life is. So if life existed, it had to exist because life had ribosome, the first cells, and this protein was essential there. So it was not the oldest one; it was one of the young ones. And this is what a part of my work at Harvard is I'm inserting different ages of this enzyme, and we're evolving these organisms. A different topic than that. That's a whole new, whole different subject. But it doesn't function well inside the modern organisms, and it causes the growth of the modern cell to be deprived. So they're very sick upon integration of these ancient genes. Keep in mind, I'm putting them inside the genome, so they're not like Rubisco's where I just work in the test tube. We are talking genome level engineering.
So ancient residues must be like organisms.
Can you?
Oh, with old genes, there must be a lot of broken fragments, and then you have to figure out how they. I mean, you found one gene.
Yes.
There must have been others around in that organism, but maybe they're all fragmented.
Yeah. So for this kind of work, we do not pick genes that once we align them, if we see gaps. So some organisms have a different version of that gene, for instance. So if we see gaps in our proteins, we cannot reliably reconstruct their paths. So we're limited as to our first alignment. I'm not extracting DNA from rocks. I am inferring them based on the DNA sequences that I have. So it's all computational in the beginning. We can't find a 700 million year old intact DNA. But this kind of methodology could actually help fill in the gaps of the other studies. And I think you're asking more human evolution kind of work, like Svante Pabo style, that's actually extracting DNA from ancient. Yeah, this can definitely help filling in the gaps. But here we're starting from complete bioinformatics level.
what kind of problems do you want to solve, maybe in 10 or 20 years, using the research you are doing right now?
That's a very good question. I want to create, so I'm very interested in ancient phenotypes, ancient behavior. So I do want to start this research paradigm, which I think is currently missing, that is dedicated completely on understanding how the ancient molecules behaved. And I do not want to just stay with proteins alone, but I want to create ancient pathways that function in the current organisms, and also ancient cells. So if you're familiar with the synthetic cells that we have, for example, I would very much be happy if 30 years from now I come here and present you guys on the ancient cell that dated, say, 2.5 billion years and how it behaved. So that's what I want to dedicate my life to, actually.
Thank you.
Help me understand something about C13 to C12 ratio. Is the selective uptake of C13 conserved with the species and through time? And then the other piece of my question is, carbon-14 is only good up to a certain time of this case. What is the decay rate like for C13? How far back in fossil records is it reliable?
To answer your first question, it is different from organism to organism. And it does change as we go back in time. So that's the whole punchline of this work as well, because we have the data set that geologists create using this isotope fractionation values. I'm not very familiar with the actual values, but I know that there are plots that shows the fluctuations. So the first goal that we have right now is to get to the youngest Rubisco and then compare the isotope fractionation value of that to what geologists find. And as far as I understand, talking to geologists, even a small fractionation difference makes a big difference in their calculations.
When you go looking kind of into the past of these proteins, do you find things that are significantly different? Like you said, the elongation factor has served its function for as long as life has been around, but how like the metabolic pathways for amino acid biogenesis, have those kind of changed over time?
Yeah, so there are a few examples that connect the ancient proteins with the ancient environments. And based on these studies, we find that based on the behavior of the ancient proteins, the older life is more likely to be hot compared to the present in terms of weather condition. And as well as the acidicity of the ancient oceans is thought to be also pretty high. And those informations come from studies that, for example, thioredoxins. The study resurrected ancient thioredoxins and mapped the properties of these thioredoxins to the pH levels. And what they see is that as they go further back in time, and thioredoxin is also proposed to be an old enzyme, as old as 3 billion years, they find that suggestively that ancient life was acidic. So these are the two environmental information that we extract using ancient reconstruction.
So you mentioned about the top-down and bottom-up approaches.
Yes.
And I'm familiar with Jack Shostak is working on the bottom-up.
Yeah.
Trying to form these like protocells and all that. I was wondering like how much is the gap between the two, like these bottom-up approaches and top-down approaches? Do they ever converge, or do you see them ever converging and getting into the same intersection at some point?
That's a good question. And what he's referring to is a study of Jack Shostak here at Harvard Medical School, where he designs these protocells, basically capsules, that will host first molecules, or first replication of molecules. So first he designs these protein cell membranes that are made of fat, lipoproteins. And then he inserts replicators, so DNA that can replicate itself. So the way we define life, is, at least as far as NASA, is that life is a system that is capable of exhibiting Darwinian selection. It's a self-sustaining system capable of exhibiting Darwinian selection. So it needs to be self-sustaining, right? So the DNA needs to replicate. It also needs to do this enough to keep its life going. So in that sense, by starting a non-life molecule, the nucleic acid alone is not life. But if you make it replicate and self-sustain it within these capsules that he designs, then you have life. So that's now becomes a bit more biological. And we are far from filling that gap from the top down and bottom up, because like scientists like me, we start from biology that we see, which is a lot more complex and complicated, because this is the biology is. But studies from bottom up start very, very in a reduced manner. But the goal is eventually merge, not just, like the Miller-Urey experiment that's produced amino acids from chemicals that propose that the amino acids that we have right now may have coming from meteorites, for example. The eventual goal is not just to make amino acids, but design cells that can replicate, produce, and have a self-sustaining life. So that will be then biology's issue once we get there.
Let's give a round of applause for Dr. Kacar.
There's much more ambiguity in natural language than we think. And the point is that most of the time we're not aware of it. There's just this black matter of often hundreds of possible interpretations of sentences that are licit interpretations of the sentences, but no one ever thinks of them.